Mitochondrial NAD+ is the currency that gets the most attention in longevity research, and for good reason. Mitochondria, the organelles responsible for generating the ATP that powers virtually every energy-demanding process in the body, are both the primary consumers of NAD+ and the primary sites where NAD+ decline with aging has the most consequential effects. When mitochondrial NAD+ falls, energy production becomes less efficient, reactive oxygen species accumulate, and the mitochondrial quality control processes that normally clear damaged organelles begin to fail. Restoring mitochondrial NAD+ is a legitimate therapeutic target. The debate is whether NMN's pathway reaches this target more effectively than niacin, and whether delivery innovations can level the playing field. NMN's marketing has leaned heavily on the idea of subcellular targeting, the suggestion that NMN reaches mitochondria more directly or more efficiently than older NAD+ precursors. The mechanistic argument involves a transporter called SLC12A8, identified in mouse research, that appeared to import NMN directly into intestinal cells. This generated significant excitement. However, subsequent research has questioned the transporter's significance in human physiology, and the broader claim of superior mitochondrial delivery in humans lacks definitive supporting evidence. The mitochondrial targeting story, as marketed, is built on a foundation that is still being actively contested in the scientific literature. What's underexplored is whether delivery innovations applied to niacin could produce comparable or superior mitochondrial NAD+ enhancement at a fraction of NMN's cost. Liposomal formulations encapsulate compounds in lipid spheres that mimic biological membranes, potentially improving absorption and cellular penetration by facilitating merger with cell membranes rather than requiring active transport. Liposomal vitamin C, liposomal glutathione, and liposomal curcumin have all shown absorption advantages in research settings. Liposomal niacin, while not extensively studied, is mechanistically plausible as a delivery enhancement strategy. Commercial liposomal niacin products exist, and the cost premium over standard niacin, while real, is dramatically smaller than the gap between niacin and NMN. Sublingual delivery of nicotinamide, niacin's closely related cousin that crosses the blood-brain barrier efficiently, is another underexplored option. Bypassing first-pass hepatic metabolism through sublingual absorption could meaningfully increase the proportion of the dose that reaches systemic circulation intact, improving both bioavailability and the fraction available for mitochondrial NAD+ synthesis. Extended-release niacin formulations, which spread absorption over several hours, sustain NAD+ precursor availability in the bloodstream over longer periods, which may more closely approximate the continuous supply that mitochondria require for sustained NAD+ synthesis than a single large-dose hit. The honest assessment is that the mitochondrial targeting advantage claimed for NMN has not been demonstrated conclusively in humans, while delivery enhancements to niacin that could address its known bioavailability limitations remain largely uninvestigated at scale. Research and commercial investment have flowed toward NMN because NMN can be patented and niacin cannot, not because the evidence clearly indicates NMN is reaching mitochondria with superior efficiency. For consumers and researchers willing to think outside the marketing framework, liposomal or sublingual niacin derivatives represent a scientifically plausible middle path worth investigating seriously.